On behalf of OCT Clinical, allow me to extend a warm welcome to you. Thank you for joining us. Our today's webinar will be about estimates for time to event data. Specifically, we will focus on endpoints typical for clinical trials in oncology. We expect the session to last about 45 minutes, followed by a question and answer session. At this time, all participants are in listen-only mode. We encourage you to ask questions at any time during the presentation, and the questions will be addressed at the end. To ask a question, simply type it in the area provided and press enter. Let me briefly introduce myself. My name is Kristina Bondareva. I'm head of biostatistics department at OCT Clinical. I'm happy to be here today and discuss with you the application of estimates to the time to event endpoints in an oncology clinical trial setting. And let's take a look at our agenda. We will first say a couple of words about the ICH E9R1 addendum on estimates and sensitivity analysis. We will then review some special characteristics of time to event data, due to which we decided to dedicate a separate webinar to this kind of endpoints. We will discuss endpoints typically used in oncology studies and provide examples of intercurrent events, which should usually be taken into account, along with strategies to account for such events. We will review possible uses of principal stratum strategy and discuss difficulties in estimating overall survival arising from treatment switching in terms of estimates. We will show how to distinguish between sensitivity and supplementary analyses. And finally, we will provide an example in diffuse large B-cell lymphoma. Let us start with reviewing the current regulatory landscape. As you may know, the draft guidance on estimates and sensitivity analysis was published in 2017 as an addendum to the statistical guideline ICH E9. Subsequently, it was implemented by regulatory agencies including EMA in July 2020 and FDA in May 2021. As a consequence of that, the sponsors now need to include estimates in clinical trial protocols, at least for confirmatory studies, though it is encouraged to apply estimand thinking in other types of trials as well. At first, when draft guideline was released, one of the frequent questions asked was whether it was applicable to oncology and time to event endpoints. The examples presented in the guideline and the majority of the published papers mainly targeted longitudinal endpoints. Yet the final guideline itself provided the answer. The framework applies to any data type, including longitudinal, time to first event, and recurrent event data. Time to event endpoints are a very special class of endpoints, which are different from more well-known categorical or continuous data types, and therefore special methods and special considerations for the analysis of such data are necessary. This also involves considerations for constructing estimates and selecting suitable estimators, which will be in the focus of our today's webinar. But first, let's take a closer look at what time to event endpoint is. In order to define a time to event endpoint, one needs to specify time origin, the point at which the follow-up time starts, the event of interest, which can be a single target event or multiple events in case of a composite endpoint. And for the analysis purpose, the circumstances where survival times are censored will also need to be specified. One of the challenges specific to survival analysis is that only some individuals will have experienced the event by the end of the study, and therefore survival times will be unknown for a subset of participants. This phenomenon is called censoring, and it may occur in the following circumstances. The study participant has not yet experienced the relevant outcome, such as relapse or death, by the end of the study. The study participant is lost to follow up during the study period, or the study participant experiences a different event that makes further follow up impossible. The collection of methods used to analyze such data, including widely used Kaplan-Meier estimates of survival times, log-rank test, and Cox proportional hazard regression, is called survival analysis methods. Most of them are designed for right-censored data, though methods for interval and left-censored data are also available. For most analytic approaches, censoring is assumed to be random or non-informative, which is an assumption equivalent to implying a missing completely at random pattern. It means that probability of censoring is unrelated to the probability of the event, and patients who are censored would have had the same risk of experiencing the event as those who continue to be followed. In contrast, patients who discontinue participation in a trial for treatment-related reasons, such as perceived lack of efficacy or adverse reactions, are likely to have a different risk of experiencing the event in the future than other patients, leading to informative censoring. It is well known that informative censoring is a source of bias for estimation. If informative censoring can be considered minimal, then the potential bias is usually ignored. There are approaches that can adjust for censoring and competing risk methods. Anyway, it is always useful to compare censoring pattern across treatment arms, including the distribution of censoring reasons. Historically, the gold standard for efficacy assessment in oncology has been demonstrating that the new treatment prolongs survival. More recently, a delay in disease progression has become a common primary objective, even for phase 3 trials. Typical time to event endpoints in oncology include overall survival, the time from randomization or from the first dose in a non-randomized trial until death from any cause, time to event endpoints such as event-free survival, EFS, and disease-free survival, DFS, and progression-free survival, PFS, which are composite outcomes that assess disease control. Progression-free survival is the time from randomization to objective disease progression or death from any cause, whichever occurs first. Disease-free survival is the time from randomization to objective disease recurrence or death from any cause. Event-free survival is typically indication-specific, but not necessarily consistent in different protocols. For example, for acute myeloid leukemia, event-free survival was defined as the time from randomization to induction treatment failure, relapse for those who have induction treatment success, or death from any cause. Duration of response and time to response are typically evaluated as secondary endpoints in early-stage clinical trials when efficacy is assessed by the best overall response and presented as the overall response rate. Per the ICH E9R1 addendum, intercurrent events are events that have the potential to occur after treatment initiation and either preclude the observation of the variable of interest or affect its interpretation. Within the context of endpoints such as PFS, EFS, and DFS, possible intercurrent events include discontinuation from the treatment due to tolerability and the initiation of subsequent anticancer therapy prior to observing an event. Within the adjuvant setting, additional potential intercurrent events for consideration include non-disease-related deaths and the occurrence of second primary malignancies. And in hematologic oncology studies, start of maintenance therapy, stem cell transplantation, possible response or remission, which decides the maintenance therapy. The estimand, or the effect which needs to be estimated, is described using five attributes: the target population, the treatment, which could be individual intervention, combination of interventions, or sequence of interventions, the endpoint, in our case it's time to event variable, a description of how to account for intercurrent events, and a population level summary for the endpoint, providing the basis for comparison, for example, hazard ratio. There are five strategies outlined in the addendum, and we will now consider them one by one. Treatment policy strategy is considered to be the most closely aligned with the ITT principle. The term itself comes from the idea of evaluating the effect of the entire sequence of treatments, from the randomization and initiation of randomized treatment, and including all subsequent treatments up to the occurrence of the event or end of follow-up. In this case, the occurrence of the intercurrent event is considered irrelevant in defining the treatment effect of interest. For example, the effect on disease-free survival is estimated regardless of whether a patient starts a subsequent therapy. It also affects the treatment attribute of the estimand, since the effect on the study drug, possibly followed by subsequent therapies, is to be estimated. In order to assess treatment effect using treatment policy strategy, data should be collected and assessed until the event of interest, even if a patient starts a subsequent therapy. Composite strategy is used when intercurrent event itself is considered informative about the patient's outcome. Therefore, it is included in the endpoint definition. For example, including occurrence of second primary cancer in the definition of DFS represents a composite strategy. Applying composite strategy for a certain intercurrent event affects the variable attribute. For example, if we're interested in the endpoint time to progression and intercurrent event is death, handling death using composite strategy would change the endpoint to PFS, which is time to progression or death. Alternatively, a hypothetical scenario in which the intercurrent event would not occur could be of interest. For example, the effect on PFS if patient had no opportunity to switch to subsequent therapy prior to experiencing an event. In this case, the treatment attribute is just the study drug. The widely used censoring of time to event endpoints at the start of the subsequent therapy in the analysis is targeting a hypothetical estimate. Other methods also exist, and we will discuss them further in the presentation, but their use is associated with strong and often unverifiable assumptions. If a hypothetical estimate is targeted, data collection beyond the start of subsequent therapy is not required. However, important data on prognostic factors for experiencing an intercurrent event, for example, for initiation of new anti-cancer therapy or a switch from control to treatment arm, may need to be collected. A while-on treatment strategy only considers what happens up to the time of the intercurrent event. A common example is palliative treatment, where alleviation of symptoms prior to death is of primary importance. Do not get confused by the name of the strategy. While on treatment, it can be used for any type of intercurrent event, not only treatment discontinuation. In case of death, it could also be labeled as while alive for clarity. When while on treatment strategy is applied, intercurrent event is taken as a competing event. There are two approaches for competing risk analyses that can be applied to implement the strategy for time to event estimates: the cause specific hazards model and the subdistribution hazards model. A number of situations in oncology can be handled using while on treatment strategy. For example, death can be considered as intercurrent event competing with the event of interest when evaluating time to certain safety events or time to progression. Anything after death is not relevant to the clinical question of interest. In principal stratum strategy, the population attribute is defined by potential occurrence or non-occurrence of intercurrent event on each of the treatments being compared. One can hence define four strata as shown on the picture. For example, S00 box is the principal stratum of patients who would not experience the intercurrent event on either treatment. Generally, treatment effect in principal stratum would be estimated using a model to predict such patients from their baseline characteristics. Effect estimation in principal stratum is very different from the so-called naive analysis performed on subgroups of patients based on the observed intercurrent event occurrence. Such naive analyses would not only compare non-randomized populations, but they are also prone to immortal time bias. Though a principal stratum strategy has not been common in regulatory oncology clinical trials, its potential utility has been topic of many discussions. Let's consider some of them. Tumor growth inhibition metrics are used to predict the effects of treatment on overall survival. Achieving a specific favorable tumor metric shortly after treatment start could be considered as an intercurrent event. One could be interested in estimating the treatment effect in terms of overall survival in patients that achieve tumor response. Likewise, since biological drugs may trigger immune responses that lead to the formation of anti-drug antibodies, ADA positivity can be considered as an intercurrent event. And a relevant question of interest would be whether ADA positive patients would still benefit from the treatment. Similar question can be formulated regarding patients who have low exposure, as measured by PK concentrations. Another example is from a real study, the prostate cancer prevention trial, in which about 19,000 men were randomized to finasteride or placebo. Men randomized to finasteride had lower rates of prostate cancer. However, it was noted that among patients who developed prostate cancer, patients from finasteride arm had a statistically significantly higher risk of high-grade prostate cancer compared to those randomized to placebo. The question of interest here would be to assess the effect of finasteride on the disease severity among those men who would be diagnosed with prostate cancer regardless of their randomized treatment. We will now review one of the examples in more detail. The Toga trial was a phase 3 randomized controlled trial comparing chemotherapy versus chemotherapy plus trastuzumab in patients with gastric cancer with overexpression of HER2 protein. In a post-hoc analysis, patients in trastuzumab arm were divided into four subgroups according to the value of TRO minimum concentration at cycle 1. It was observed that patients who had lower exposure appeared to have shorter overall survival compared with other patients as indicated by the red line on the survival plot. However, due to some important differences in the baseline characteristics, it was not possible to conclude whether the differences were due to exposure or to the disease burden. A follow-up case control analysis of these data was conducted by the FDA. Patients with low trastuzumab exposure were matched for risk factors including ECOG, history of gastrectomy, ethnicity, number of metastatic sites, and level of HER2 overexpression with those from the control arm who were not receiving trastuzumab. In this analysis, the median OS was 7.7 months in the lowest trastuzumab minimum concentration quartile compared with 7.5 months in the matched control group, suggesting that trastuzumab conferred no benefit in terms of OS when minimum concentration during cycle 1 was low. These results, together with further exposure response analyses based on the Toga data, led to the initiation of a fully powered clinical trial comparing standard versus high-dose trastuzumab for the identified subgroup of patients. Although not explicitly described within the Estimand framework, this approach undertaken by the FDA implicitly targeted a principal stratum strategy, and this example illustrates how such estimand can be of clinical importance and impact a drug development program. We have reviewed the 5 strategies contemplated by the Addendum. Let's now consider one of the most controversial intercurrent events, treatment switching. The start of a new anti-cancer therapy after discontinuation of a studied treatment and prior to observing the event of interest, that is, treatment switching, has become increasingly common as more treatment options become available to patients. This, of course, complicates assessment of benefit provided by the initially randomized study treatment, especially on the overall survival. When speaking about estimating treatment effect on overall survival in presence of treatment switching, several different scenarios can be considered. In the first picture, in both patients in both test and control group can switch to a subsequent therapy in line with the clinical practice. The clinical question would be, what is the benefit on overall survival of the study drug followed by any subsequent therapy compared to control followed also by any subsequent therapy? In the second display, patients from the control arm may switch to the investigational drug, which is already an approved treatment, or is soon expected to be approved as next line of treatment. This situation also reflects clinical practice and can be handled by treatment policy strategy. The third situation is different in that a patient from the control arm may cross over to the investigational drug, which is not yet an approved therapy, or to another investigational therapy with similar mode of action. In open-label clinical trials, patients might discontinue and seek the opportunity to receive investigational product in another clinical trials, as was the case in recent studies of checkpoint inhibitors, where a lot of patients from control arm switched to other non-protocol checkpoint inhibitors outside of the clinical trial upon progression, or even discontinued once they found out that they were randomized to the control group. When there is no effective alternative for subsequent therapy, patients are often allowed to cross over to the investigated treatment arm upon progression. It could even result in similar exposure to investigational therapy in both arms. Such circumstances cannot occur outside of the clinical trial and do not reflect clinical practice. So, the valid research question would be, what would the benefit on the overall survival be if the possibility to cross over had not existed? This treatment effect would be more relevant to both patients and researchers. The hypothetical strategy can be implemented using simple censoring at the time of the crossover if the assumption of independent censoring holds. It usually does not hold, since the decision to switch is probably related to clinical worsening. Inverse probability of censoring weighting approach allows to adjust for censoring by assigning weights to the remaining observations using baseline and time-dependent covariates. Patients with similar characteristics to those who switched receive high weights. However, this approach is subject to the assumption of no unmeasured confounders. Other possible options include rank-preserving structural failure time models and two-stage models, which are based on different assumptions. The former assumes that the relative effect of treatment is the same regardless of whether it is initiated from the start, from the moment of the randomization, or from the time of the crossover. It is called common treatment effect assumption. It allows to recover which event times would have been observed had this patient not crossed over. This approach is applicable for small sample sizes, placebo-controlled or add-on studies, and does not require covariate information. Two-stage models are not based on common treatment effect assumption. They introduce a secondary baseline, often at the time of progression. The post-progression survival is compared between control group switchers and non-switchers, using an accelerated failure time model, adjusting for covariates measured at baseline and at the time of progression. The resulting slowdown factor is inversely applied to control patients who switched to adjust their survival times observed in switching. The obtained counterfactual survival times for the control group and observed times for the investigational group are used then in Cox regression model to estimate the treatment switching adjusted hazard ratio. As stated in the EMA Q&A document dedicated to this topic, which method would be more appropriate depends on which assumption seems most reasonable or robust to deviations. Historically, in oncology, sensitivity analyses were performed in more or less standard ways, as described in the FDA guideline clinical trial endpoints for the approval of non-small cell lung cancer drugs and biologics. Tables with suggested analyses of PFS for primary and for the sensitivity analyses are provided in the appendices to the document. Often, several sensitivity analyses were performed with different censoring rules, for example, by censoring at the time of treatment switching or by counting it as an event. The addendum makes a clear distinction between the analyses that target the same Estimand sensitivity analyses and those that answer different research questions and are associated with a different Estimand. Those are labeled as supplementary analyses. As shown on the picture, sensitivity analysis is an analysis that targets the same Estimand. It might use different assumptions to explore the robustness of inferences from the main estimator or deviations from its underlying modeling assumptions and limitations in the data. Let us try to guess for a few examples whether the mentioned analyses are sensitivity or supplementary. The first example. Suppose our primary estimand answers the following clinical question of interest. Does the drug improve disease-free survival if no patient had received subsequent therapy? And time to event is censored at the intercurrent event occurrence. Would the analyses, which include subsequent anti-cancer therapy as an event, or consider events regardless of subsequent therapy, be considered sensitivity analysis in the light of the addendum? And the answer is no. The primary estimand is hypothetical estimand, and the other two analyses use composite and treatment policy strategy, so they correspond to different estimates and cannot represent appropriate sensitivity analysis. Second example. Could analyses vary in the timing of events at scheduled assessments, earliest versus latest scan data, event time as assessed by the investigator, be considered sensitivity analysis? Yes, these analyses are in the spirit of the addendum sensitivity analysis definition. Third, here we see a very commonly used question of how to deal with the event registered after several missing assessments. So, could we address this question with a sensitivity analysis? And the answer could be twofold. It could be treated as a missing data problem and addressed by sensitivity analysis, or if missing assessments could be caused by an underlying intercurrent event such as COVID-19 pandemic, and in this case it should be taken into account in the estimand definition. Finally, missing data resulting from study withdrawal for safety reasons should also be considered by appropriate sensitivity analysis. The example I will present is related to chimeric antigen receptor T-cell therapy, or CAR-T for short, in diffuse large B-cell lymphoma indication. And the example is meant to describe how Estimand framework could actually help design such a study. When a patient who has been treated with rituximab, cyclophosphamide, doxorubicin, vincristine, and prednisone develops a recurrence, this patient could be treated with either standard of care, which is autologous stem cell transplantation, or he or she could receive an investigational personalized CAR-T therapy, currently approved to treat patients with diffuse large B-cell lymphoma in a later setting. Patients can experience different journeys on each treatment option. In case of standard of care, patient will initially receive salvage chemotherapy. The best standard of care therapy after the previous regimen would be high-dose chemotherapy followed by autologous stem cell transplantation for patients who achieve a deep remission. Autologous stem cell transplantation is considered the only possible curative option currently approved for such a relapsed disease. If a deep remission is not obtained after 2 or 3 cycles of first salvage chemotherapy, approximately during 6 weeks, the patient will receive a new salvage chemotherapy, still with the goal to achieve deep remission and subsequently receive transplantation. If there is no improvement of the disease on 2 salvage regimens after approximately 12 weeks, then the standard of care option would be deemed to have failed. In this situation, CAR-T therapy could be offered to this patient because it is approved in this later setting. Alternatively, the patient may receive CAR-T immediately as an experimental option in this earlier setting, also with the goal to cure the disease. Patients' T cells would be collected by leukapheresis to enable CAR-T to be manufactured. Some patients might need bridging therapy to control the disease, during the manufacturing process. Within the week before the infusion of CAR T cells, lymphodepletion chemotherapy would be given to ensure that the immune system does not destroy the infused genetically modified patient's T cells. It is possible that the patient would attain remission, or that the patient's health status would worsen, or that manufacturing would fail, and CAR T administration would be delayed or cancelled. However, tumor response or progression before CAR T infusion would not be a final outcome. If the patient did not receive CAR T therapy, he or she might still be a candidate for additional chemo. If there is no improvement of disease after approximately 12 weeks, then it would be considered that the treatment strategy of CAR T administration has failed. Such a variety of possible patients' journeys presents certain challenges to identify the adequate research question and define the treatment effect of interest for a randomized controlled trial comparing these two treatment options. First, the therapies to be compared are two treatment strategies, not simply two different treatments. Both strategies are complex and involve multiple steps and decisions, and patients may not receive all the components. Second, there is an initial period when similar treatment effects are expected from both treatment groups, since the bridging therapy administered before CAR T infusion is similar to the standard of care salvage chemotherapy. Finally, after the failure of standard of care treatment strategy, a CAR T may be administered, and the subsequent outcomes in the standard of care treatment groups would reflect also the effect of this crossover, not the effect of standard of care strategy alone. The Estimand framework helps to address these challenges and provide a precise definition of the treatment effect of interest using the 5 Estimand attributes and the language offered by the Estimand framework. So let's consider the treatment attribute first. The study could be intended to evaluate the effect of CAR T against transplantation alone, focusing on the potential curative characteristics of both options. It could be studied by randomly assigning patients to study arms only when they are already prepared for both the administration of CAR T after its successful manufacturing or to the transplantation after reaching a sufficient remission on salvage chemotherapy. All necessary preparatory steps would then be a part of a common run-in phase. However, the population for the comparison would then be only a subset of the population initially considered eligible to receive CAR T or standard of care. It is likely that some eligible patients would not reach the point of random assignment and therefore would not contribute to the assessment of treatment effect. Patients excluded due to insufficient remission could still benefit from CAR T therapy, but this would not be reflected in the estimated effect. This would not address the following scientific question of interest. What is the relative clinical benefit across the entire patient journey once a CAR T or standard of care treatment strategies are prescribed? Therefore, it seems of interest to compare the entire treatment strategies instead. This also is in agreement with clinical practice, where not all the patients will receive all the components of the treatment. Consistent with the definition of the treatment, the population of interest includes all patients eligible to receive either CAR T or standard of care treatment strategies as specified by inclusion and exclusion criteria. Typically, in this disease setting, the primary variable or endpoint is event-free survival. the time from random assignment to the failure of treatment strategy. Here, failure is defined as first documented disease progression, stable disease, or death from any cause. Note that disease progression or stable disease are only considered as failure if observed at or after the week 12 assessment. Tumor assessment after the first course of bridging or salvage chemotherapy do not determine the outcome of the treatment strategy and are used only for decision making. The most exciting part of, defining, describing an estimand is identifying the intercurrent events and selecting strategies. Three intercurrent events could be identified. The first is manufacturing failure in the CAR-T arm or failing to receive autologous stem cell transplantation in the standard of care arm. Following the treatment policy strategy, such events should be ignored, and patients should still be observed until the final event of interest. The second is new anti-cancer therapy started before observing the event. If a patient takes an unplanned anti-cancer therapy, it would be difficult to interpret the outcome as reflecting the effect of that treatment strategy. This intercurrent event could then be handled using a hypothetical strategy, evaluating the effect had patients not been offered such an alternative treatment. The third is insufficient remission, stable disease or progressive disease at week 6. As already discussed, intermediate disease assessments do not determine the final outcome. They are ignored following the treatment policy strategy, as reflected in the variable definition. The typical summary measure for comparing event-free survival is hazard ratio, though in this case it could be noted that proportional hazards assumption will likely to be violated since similar treatment effects are expected on bridging and salvage therapies in the initial phase. In addition, both CAR-T and transplantation are potentially curative, and there will be a proportion of patients who are cured and might have similar outcomes. Alternative summary measures to quantify treatment effects in presence of these non-proportional hazards are still being investigated. The resulting schematic presentation of the treatment effect of interest is provided on the slide. We have reviewed some of the aspects of oncology trials and time-to-event analysis in the light of the Estimand framework. As perceived, it does not change analysis methods common in cancer research. Instead, it facilitates discussions between stakeholders to ensure alignment between the key question of interest, study design, analysis, and interpretation. Some useful references are provided on this and the following slide. You will receive the slides after the presentation, so I hope this list will be helpful. And now we will proceed to address the questions. So the first question is, do you know which estimands were used in polaric study in diffuse large B-cell lymphoma? Yes, I've heard of this study. It is having a strong impact since for the first time in 20 years, at least some improvement on PFS over R-CHOP as the first line of treatment of diffuse large B-cell lymphoma has been demonstrated in that study. This month, the ODAC meeting was conducted, and I have seen parts of the recording and also the briefing document. I didn't go into details, to be honest, but my impression was that initially Estimand language was not used when the study was planned. Its primary endpoint was PFS, and the primary analysis, if I'm not mistaken, did not censor for new anti-cancer therapy. Therefore, implicitly, we can say that it used treatment policy strategy for this intercurrent event. But the FDA reviewers were in disagreement with such strategy and said that they usually censor for such events for this indication. So, in a way, they would prefer to see a hypothetical estimate. They conducted their own analyses, and the whole description of the situation actually reminded me of a well-known example in diabetes, which is commonly used to demonstrate the usefulness of the estimate framework for discussing the treatment effect of interest with the regulators, especially. And I think this study would have also gained from applying this concept from the start, from the definition of the effect of interest in the protocol using the estimate framework. I hope this answers the question. So the next question is how do you select a strategy? Well, this one requires a lot of time to answer. Actually, there's no one correct answer to that question. We have reviewed the 5 strategies today, which are outlined in the addendum. While on treatment and principal stratum would hardly be used for a primary estimate, we have mentioned some possible uses of while on treatment for safety analysis and principal stratum as an enhanced way while on treatment for safety analysis, yes, and the principal stratum as a better way of subgroup analysis. Um, Composite strategy is directly linked with a variable attribute and which events are included in the definition of composite variables such as event-free survival or disease-free survival for each indication actually requires harmonization. Now it's not being done consistently. There's data can initiative which already works towards achieving this harmonization. So in most cases, the most difficult choice would be between treatment policy and hypothetical strategy. The addendum does not express preference for one or for the other. So the choice would depend on the specifics of the disease, the mechanism of action of the drug, and current treatment landscape. Uh, possible treatment options for the patients. For treatment switching, do discuss with the study team, with the investigators, whether patients may switch to another investigational treatment with the same mechanism of action, as was the case in checkpoint inhibitors. This type of confounding would be better avoided, uh, maybe by providing alternatives within the given protocol. Yeah, I do not see other questions for now. So if you happen to have another question, oh, I now see, yeah, I can see another one. Are methods like rank preserving structure failure time model implemented in statistical software? If I'm not mistaken, it is not implemented as a built-in option in SAS, um, but this method is available in ARP and Stata packages. Yeah, uh, I think SAS doesn't have this built-in option for now. Yeah, now I think we have addressed all the questions. Uh, Thank you very much for your attention. Uh, the final recording will be available on the OCT YouTube channel. And if you're interested in estimates and would like to dive a little deeper, you could also find our previous webinar on ICH9R1 Addendum on our YouTube channel. And we will provide the slides of today's presentation and address any questions that you might have, and you can still type some questions or send us your questions, and we will address them in a follow-up email. So thanks once again for joining, and have a great rest of the day. Bye.